Hello, welcome to 13 Universal Learning Beyond Barriers, and this is A-level CIE Biology with schedule. So we're going to be starting a new series today, Cell Membrane and Transport Series 1. If you have been following our videos, we have done a lot of series on cell structure, on biological molecule, enzymes, and then we're starting cell membrane and transport. Please follow us, okay? Like the video, share it, comment, and of course subscribe. So let's go into the question. The first question says, read with me. It says the fluidity of the cell surface membrane can be changed by a number of factors. So as the fluidity of cell surface membranes decreases, which process would be least changed? Which process would be least changed? Okay, let's go. One is active transport. Two, that's B, diffusion. C, endocytosis, and then osmosis. Guys, if you remember that the fluidity of surface membrane has a lot to do with phospholipid bilayer. Remember the phospholipid bilayer? Now it just tells us that fluidity tells us, it just tells us that the phospholipid molecules move around. Okay, they move around within their monolayers. You know phospholipid molecule, something like this. So this is another phospholipid molecule. So when we mention fluidity, it has to do with a lot more with phospholipid bilayer. Okay. So let's go straight to the question. So this is phospholipid bilayer. Now the next thing we're going to ask ourselves is which of these processes require membrane, phospholipid bilayer? Which of these processes require the route, molecules passing through this membrane? Okay. Now active transport is the movement of polar molecules or ions from low concentration to high concentration, that is against concentration gradient. And don't forget that we are transporting polar molecules here, so it's not going to pass through phospholipid membrane, it's going to pass through carrier protein. Is that okay? So carrier protein will be is involved in active transport. Diffusion, all right, simple diffusion passes through phospholipid membrane. Endocytosis also involves phospholipid bilayer. Osmosis also involves it. All right. The one that is least affected is active transport because it involves solely protein molecules. Okay, fluidity deals with mainly phospholipid molecules. Okay, the one that involves only protein mostly is active transport. So that's the least affected. What do you think? A. Let's go to the next question. Number 2. It says which molecules in cell surface membranes contribute to cell recognition? Let's go. That's A, B, C, D. So cell recognition. A says cholesterol. No, cholesterol is not involved in cell recognition. Cholesterol and phospholipid, they prevent passage of polar molecules. They regulate fluidity of the membrane. Okay, and then they give mechanical stability to the membrane. Okay, so they're not involved in cell recognition. Or anyway, see two of them. The options that have cholesterol or phospholipid, they are not supposed to be part of the answer. So this is C. C says glycolipids, all right, can act as a marker, or it can act as a tag, a label, all right, an antigen. The same with glycoprotein, the same with protein. Okay, so three of them are involved in cell recognition. Glycoprotein is involved in cell recognition and cell adhesion. That's glycolipid, cell recognition, cell adhesion. Glycoprotein is involved in cell recognition, cell adhesion, cell signaling. All right, protein is involved with cell recognition, cell adhesion, cell signaling, transport, and enzymes. All right, so but all of them play a role in cell recognition. Okay, so that is why the answer is C. Okay, glycolipids. All right, which is correct for facilitated diffusion and active transport? Emphasize. right? Look at that. And so it must be an intercept. It must agree for facilitated diffusion and active transport. A says both depend on solubility of the transported molecule in the lipid bilayer. They don't depend on solubility. Okay. All right. Of course, they are transporting polar molecules. All right, and this is talking about non-polar molecules. It's only non-polar molecules that dissolve in the lipid bilayer. So facilitated diffusion and active transport are involved in transport of ions and polar molecules. So A is wrong. Okay. B says both increase as concentration of transported molecules increases. Well, that could affect facilitated diffusion, but not active transport. Active transport is not affected by concentration gradient. As a matter of fact, active transport sets up concentration gradient. Is that okay? Yeah. So that B is only for facilitated diffusion. C says both require the use of ATP. Well, no, it is only active transport that, all right, requires ATP, all right, because it is a movement of polar molecules from low concentration to high concentration, low to high, and that is against concentration gradient. So it requires ATP. But facilitated diffusion is a movement of polar molecules or ions from high concentration to low concentration. So that's down concentration gradient. Okay, so that is why C is not the answer. So most likely D looks like the answer. Both require the use of membrane proteins. Yes, both of them transport polar and charged molecules, and they know that phospholipid bilayer and cholesterol, they repel polar molecules. So polar molecules can only pass through transport proteins, which are channel proteins and carrier proteins. So the answer here is delta D. Okay, D. Let's go to the next question. Number 4. Which features increase the efficiency of ion uptake by a root hair cell? Increase the efficiency. All right. Many mitochondria in the cell, high concentration of ions in the vacuole, protein carriers in the cell surface membrane. Okay, which one? Pause the video and tell us the answer. So which features increase the efficiency? Remember, the emphasis is on efficiency of the ion uptake by root hair. Now, ion uptake by the root hair is by active transport. Okay, ions move from low concentration in the soil into the root hair cell. Okay, from the low concentration in the soil to high concentration, all right, in the root hair cell. So ion uptake is by active transport. All right, so we are dealing with active transport here. Number 1, active transport requires, it requires a lot of energy because I'm moving against concentration gradient. But you see, we're talking about efficiency, not just to start it. All right, so number 1 says many mitochondria in the cell. Now, many mitochondria will increase the efficiency. If you have said mitochondria, yeah, mitochondria is needed, all right, to start the process. But many mitochondria, that means if you increase the number, as the number of mitochondria increases, then the efficiency of ion uptake also increases. Do you get that? So 1 is correct. 2 says high concentration of ions in the vacuole. Remember that concentration gradient does not affect active transport. This is on active transport, okay, because ion uptake is by active transport, all right? So it affects facilitated diffusion, but not active transport. So 2 is wrong. 3 says protein carriers in the cell surface membrane. Now, protein carriers in the cell surface membrane, that makes a lot of sense, okay? Protein carrier, all right, is involved in active transport, but however, we are talking about efficiency. So how do you increase the rate of active transport? Is by increasing the number of protein carriers. So if they have added here the increasing number of protein carriers, this would have been suitable for this efficiency. So, but they said protein carriers, all right, that's not suitable for efficiency. Increasing the high rate of it, you have to put in the number, okay, or many of these. So because of that, it's only number 1, many mitochondria in the cell, that is more suitable option to the question. Okay, that answers the question. So the answer is D, number 1. Please, if you have questions, chat us on our okay, or YouTube, okay, and then ask your questions. But you can replay the video to understand more. All right, let's go to the next question. The diagram shows the fluid mosaic model of a section of a cell surface membrane. That's it. Okay. Yeah, the fluid mosaic model. I hope you still remember the fluid mosaic. Fluid means that the phospholipid molecule, these are the phospholipid molecule. The phospholipid molecules, okay, they move around, and even the proteins, okay, they move around within their monolayer. Mosaic means that the arrangement of protein molecules, the protein molecules are arranged in a scattered. So can you see they are scattered everywhere? That's mosaic. Then I said, what is the role of each of the component labeled 1 to 3? 1 is glycolipid. Can you see? This is glucose chain, all right, attached to phospholipid. Glucose chain is glyco. Phospholipid is lipid. So that's glycolipid. Number 1 is glycolipid. Remember, I told you that glycolipid has two main functions. One, it acts as antigen during cell recognition. Secondly, it helps in cell adhesion. So let's check where it acts as antigen here. Okay, A says acts as antigen. It's only A that says acts as antigen. All right. Okay, but it says stabilizes the membrane. It can stabilize the membrane. It can stabilize because this forms hydrogen bond. The glucose chain here forms hydrogen bond with water and it's surrounded. Okay, and that could stabilize the membrane. All right, but the major function is A. All right, but let's check again. Number 2, C2. Can you see the structure 2? Structure 2 is glycoprotein. Anytime you see a rectangle, all right, or whatever square, like these structures are protein. This is protein, this is protein, protein, this is channel protein. All right, that's protein, protein, that's channel protein, that's protein. But if you see a protein attached to glucose chain, that's glycoprotein. Glycoprotein has three functions. Number 1, it acts as antigen for cell recognition. Number 2, glycoproteins is involved in cell signaling. And number 3, it helps in cell adhesion. So chemical receptors for cell signaling. Okay, so if you check it, and D, this controls fluidity. Now that makes D wrong. It doesn't control fluidity of the membrane. What controls fluidity of the membrane is phospholipid bilayer. Can you see that? And cholesterol. So that makes a most likely the answer. Number 3, see number 3 here. This is phospholipid molecule. That's 3. So allow lipid soluble molecules to pass through antigen. Yes. Yeah, it allows only lipid soluble to pass. It does not allow water soluble. So it repairs water soluble molecules, but it allows lipid soluble. So the answer is likely the option A. All right. So let's go to the next question. All right, number 6 is quite a big read. Okay, long read. Okay, long read. Let's go on. Let's go on. It says proteins in the cell surface membranes of human cells and mouse cells were labeled with red and green fluorescent dyes, respectively. When a human cell and a mouse cell were fused together, the red and green fluorescent labels were at first found in a different region of the cell surface membrane. Different region. Then what happened? Of the hybrid cell. But after 40 minutes, yes, they were evenly distributed in the entire cell surface. Can you see that surface membrane? Can you see that proteins move around? So they are fluid. Fluid means moving around. What explains this observation? Because they move, it shows that they are fluid. All right, it says all protein molecules in the cell surface membranes are fixed. No, they're not fixed. If they were fixed, they wouldn't have been evenly distributed. They were at a different region, but they were evenly distributed in the entire surface, okay, membrane. So that eliminates that option A. Option B says groups of protein and phospholipid molecules in the cell surface membrane are attached to each other and move together. No, they are not attached to each other and they don't move together. All right, it's only protein that move here. They move, both of them move, but they're not attached to each other. Okay, both proteins and the phospholipid molecules, they move, but they are not attached to each other. Okay, all right. So C says only protein molecules in the outer layer of the cell surface membrane can move freely between phospholipid molecules. No, not only the ones outer. Okay, because you have inner layer and outer layer. This says protein molecules in the outer layer of the cell surface membrane and those which spans the bilayer can move freely between phospholipid molecules. So this is correct. So whether they are in the outer layer or the inner layer, they all move freely. Okay, so the answer is delta. Okay, 7 says the diagram shows a plant cell. You can see that. That's a plant cell. That's a plant cell. So the plant cell is put into a solution with a water potential less negative than the cell content. This is very important. A water potential less negative than the cell contents. So what will happen to the appearance of the cells? Okay. Please, before we go on, when they say less negative, what does it mean? Tell me. Yes, what does it mean? Less negative means higher water potential. Okay, that word less negative. Okay, it means it has high water potential. This is of course the this thing for the symbol for water potential, this is water potential. So less negative means it has high water potential. Okay, so the water potential here is high, quite high, and it is higher than the cell content. Okay, you see it. So how will water move? So inside the solution, the water potential is high, but in the cell content, all right, watch, this is water potential, this is still water potential. Okay, one is in the solution, the other one is in the cell content. Do you get? So in the cell content, the water potential is low because that is higher. So how does water move? Water will move from high water potential to what? Low water potential. So water will move from the solution into the cell content. And you know, when water moves, what happens? When water gets into the cell, what happens to the cell? Okay, which one? Is it A? What do you think? Will the cell shrink? No, the cell is gaining water, it will not shrink. Okay, this is smaller, and I don't think these two cells are not the same size. Okay, will the cell plasmolyze? Yeah, plasmolyze means it's losing water. This plasmolysis. Okay, the cell membrane is detaching from the cell wall. Can you see cell membrane here is detaching? So that means this water has lost a lot of this. Sorry, this cell has lost a lot of water. Okay, but in this case, water is entering the cell. Okay, it's not leaving the cell. So see, the cell here is turgid. Can you see? It's turgid. It's turgid. All right, and then the cell is also turgid, but it's very big. Okay, so in the actual past paper, this, all right, cell is like this. Okay, that means that it's the same size. Okay, not, you know, in this size, this is bigger, but you know, so the cell is going to become turgid because water will enter into the cell from the solution to the cell by osmosis, and due to sugar pressure the cell becomes bigger. So this is the actual cell, all right, and that is D. No, that is C. Okay, all right, can chat us up. Now look at this. The diagram shows part of a cell surface membrane. This is a cell surface membrane here. Yep, let's look at it. Which molecules have both hydrophobic, that's phobic, not phobia, phobic and hydrophilic regions? All right, pause this video and try. What is hydrophobic? Hydrophobic means water hating. All right, structures that withdraw from water. They don't come in, they don't form hydrogen bond with water. Hydrophilic is water loving. Filio, hydrophobia, afraid of water, or the hydrophobic, this interacts with water. It points towards, this always find towards water, this found withdrawn from water. So let's go on. Number one is phospholipid. This is a phospholipid molecule. This is the phosphate head, can you see it, and the hydrophobic tail. Okay, so you can see that hydrophilic head, this is phosphate head, which is hydrophilic, it points towards water. Hydrophobic withdraws from water. So number one has both hydrophilic, this phospholipid one has both hydrophobic and hydrophilic regions. So look for option that has one. Okay, we eliminate D. So we're looking at A, B, C. All right, number two. Okay, you can see number two is this protein. Number two, this protein, this part of this protein is facing towards water. this part here is hydro. So the one that's facing towards water is hydrophilic. These ones that are embedded within the bilayer, phospholipid bilayer, is hydrophobic, and even this part is also hydrophobic from here because they are embedded inside the hydrophobic core. So it has hydrophilic, which is this part, and hydrophobic. So 2 is correct. Let's look at the option that has 2. Okay, we have limited. This doesn't have 1. This is limited. This is limited. So we narrow it down to A and B. Now 3 is the glucose chain. Can you see it? That makes this entire structure glycoprotein. This glucose chain form hydrogen bonds with water, okay, but is not embedded inside the membrane, so it's entirely hydrophilic because it's facing water. The glucose chain is hydrophilic, it's not hydrophobic, so that makes us to eliminate 3. 3 cannot be the answer. But if you check 4, so the answer is A. But let's look at it. This part of this number 4, this is a channel protein. This is a hydrophilic channel. Okay, molecules pass through here, right? This hydrophilic channel. Now this part is facing water. That part water. This part here water. But the ones within the hydrophobic core is hydrophobic. So it has both hydrophobic and hydrophilic. So structures 1, 2, 4, and 5. Let's call it 4, 5. 5 is also, this part is facing water. That part is not facing water. That's hydrophobic. So 1, 4, 1, 2, 4, and 5 are the answer. Okay. All right. So look at number 9. One type of antigen is formed by a reaction between two different molecules. Antigen. Okay. Remember that glycolipid is an antigen as well as protein. All right. It is two different molecules. That means it has maybe lipid and carbohydrate. All right. Or lipid or carbohydrate and protein. So 2. This, apart from oxygen, which other elements are found in this antigen? Which other elements? So which other elements? What elements make up lipids, and of course protein and carbs? Okay, let's look at them one by one. So this is already having oxygen, carbon. Now if this is lipid will have carbon, hydrogen, oxygen, plus carbohydrate, carbon, hydrogen, and oxygen. Okay. Now this says B. B does not have carbon. Carbon must be there, right? Now phosphorus shouldn't be there because glycoprotein doesn't have phosphorus. Okay. So this is eliminated. All right. It's only nucleic acid that have it. Okay. D says carbon, hydrogen, nitrogen, and sulfur. No. Of course, we're looking at this sulfur because we are looking at glycolipid. Okay, glycolipid has carbon, hydrogen, oxygen. Okay, and if you say this is glycoprotein, okay, this sulfur is not always in protein. This sulfur is not always in protein. Okay, all right, so the major element in protein is carbon, hydrogen, oxygen, and nitrogen. All right, so the most stable answer here, apart from oxygen, we should have carbon and hydrogen plus C, because these 3, carbon, hydrogen, and oxygen, they are all found in both lipid and carbohydrate, and lipid and carbohydrate can form an antigen, glycolipid. All right, so this is why we chose 9. The answer should be A only. What are the features of oxidative diffusion? Okay, it uses protein channels in the membrane and is driven by energy from ATP. No, it doesn't use energy from ATP. It doesn't use energy from ATP. All right, it moves molecules. You know what? Pause this. See, I want you to try this question, please. Try it and send me the answer. Okay, send me your answer right now on the YouTube comment section. Send me the answer. What is the answer to this number 10? Okay, try it. Okay, so it uses protein. Now for cell diffusion, they don't use ATP as well, because they're moving down concentration gradient. They use their own kinetic energy. And 2, it moves molecules from regions of high concentration to lower concentration. That's correct. and is driven by kinetic energy of the molecules which are diffusing. This is correct. So this is correct about state diffusion. High to low, driven by kinetic energy. Number 3 says it uses protein channels, yes, in the membrane, and maximum rate of diffusion depends on the number of, yes. So yes, it uses protein channels, channel protein. So as the number of channel protein increases, then more molecules will pass through, okay, them, and they will have higher rate of diffusion. So number 2 and number 3 are correct. Okay, the answer is C. All right. So hope you enjoyed this class. Sorry for the background noise. Okay. Thank you so much. I want you to leave your comment question sessions, I know, at the comment sections, and then please do like, subscribe, and share our videos. Okay. So we're going to be bringing more videos from other subjects, experts too. So thank you for watching. See you in the next series too. Bye.